Before we understand the entire chapter of biotechnology and its principles, chapter 11 of grade 12 textbook biology, we need to know we need to have an overview of the entire process of biotechnology. What all are the steps involved in it? So first and foremost, if you look at biotechnology, there are two tools that will come under it. The first one takes care of everything called as genetic engineering, which is engineering the genetic material or changing or altering the genetic material. The second one is called as bioprocess engineering, wherein all that you require for maintaining or changing the genetic material, that is, maintaining a sterile environment for genetic engineering, comes under bioprocess engineering. All right. So let us have a look at which all are the steps that will be followed for genetic engineering. First one is called as identifying the gene of interest. So what is meant by a gene of interest? It could be any gene that I need in another organism to produce some product, or it could be any gene that I have identified to be put in another organism so that it gives me a particular trait or particular product. Okay, so let us say I have identified a gene called as antibiotic resistance gene, and this is found in one organism, let's say bacteria, in its genetic material. This is the genetic material of the bacteria, inside which I have my gene of interest. This is the bacteria. This is the bacteria in which it has a genetic material. Inside the genetic material of the bacteria is your gene of interest called as right now, the example that we have taken is antibiotic resistance gene. Okay. So what do I do after I identify my gene of interest? I need to take out this entire DNA. That is called as DNA extraction. I cannot take out only the gene of interest. I'm extracting the entire DNA. Probably you have done this process inside in your lab practical called as isolation of DNA from papaya or any leaf you must have done in the lab. Same process, but here we have to do a refined process. So steps we will be learning in detail. Okay. So first thing, identification of gene of interest. Here, example we have taken is antibiotic resistance gene. Then, once that is identified, the entire organism we are going to select and we are going to isolate or extract the entire genetic material, the DNA of it. After that is done, I have this here out, okay, in a test tube, let's say. Inside that is my gene of interest over here. What do I do next? I need to cut this out. How do I cut it? Using scissors, I'm going to cut. But I don't have a scissor right now which can cut a DNA, right? So I need to use something called as molecular scissors, which are enzymes. What kind of enzymes are they? They are called as restriction enzymes, about which you are going to learn in detail. All right. So these are restriction enzymes. What do? They cut out our gene of interest. They not only cut out the gene of interest, they cut this entire DNA that you have extracted into tiny, tiny fragments. So I'm going to make it like this. All right. These are all the fragments, different fragments that I have. After what process? After I added the restriction enzyme to the extracted DNA. I'll repeat from the beginning. You have a gene here that is of interest to you, meaning you want it for genetic engineering process. So what did you do first? First, you identified that gene. Then you extracted the entire DNA from that organism, which is a bacteria in this case, right? So you have that entire DNA with your gene of interest in it. Next step, what do? You have to cut this out. For that, we use some enzymes called as restriction enzymes. They not only cut the gene of interest, they cut the entire genome or the genetic material. In this, when you cut, you get different fragments, and one of those fragments will have your gene of interest, right? Like this. So this part, it has cut out. The restriction enzyme has cut out. How to take it out? You can't pick it up with your hand. So what do? You use something called as a gel electrophoresis, for which I'll just show you how the picture goes. But all this, every single process we are going to learn in detail, all right? So don't worry about what is happening. If you're not following, don't worry. Just understand the process right now. So we have, what we have? We have in a test tube, our genetic material. Inside that we have our gene of interest. We have added the restriction enzyme. So this entire mass is cut, tiny, tiny pieces it is right now. Right? I'm going to cut it and then show like this. You have, right? So what do? We pour it into these wells of this unit. This unit is called as gel electrophoresis unit, about which I will teach you. Don't worry. Go back. Once I put it, what happens to it also I'll tell you. Otherwise, the story will not be complete, right? So the DNA you have added into all these holes or pores called as wells will start moving. Why is it going to move? We are going to learn. Okay. So once it starts moving, at one particular length will be our gene of interest, which I can cut out like so. Okay. And that process called as elution, which we are learning, right? So you have our gene of interest. How many copy do we have? We have only one. Is that enough? It's not enough for us. Okay, we are going to do some huge processes in the genetic engineering. We are going to do. We cannot have only one piece of DNA and then say, oh, I want to do genetic engineering. Not possible. So what will we do? We have to make multiple copies of that same gene. I have this gene right now. After this process of gel electrophoresis, I have only one gene that I have to make multiple copies. X, 100 times. So for that, we use a process called as PCR, polymerized chain reaction. So into this polymerized chain reaction unit, when you put our one gene of interest, we will get that same gene, multiple copies, many copies. Yeah. So for what we are using PCR, we are just using the PCR unit to make multiple copies of that single gene, that gene of interest, which was in this case antibiotic resistance gene. Clear? Okay, I hope. Let's see. All right. Okay, I have that gene. Now what do I do with it? I need to put it inside another organism. Okay, I'm going to use another color. This is my bacteria number 2. It may not be always bacteria. It could be plant. It could be animal, about which again we are going to learn. All right. So this gene, I have many copies. I'm going to put it inside this host. How many hosts will I take? One host, but multiple copies of the same host. Like bacteria, if I'm taking, I cannot take, oh, come one bacteria, I'll make recombinant out of it. No, not possible. Okay. I need to have a colony of it. I need to have a lot of it. So, but then when we show it in picture, we're just going to show in one. Right. So I have my host in multiple copies. Into this, I am going to add my gene. How am I going to add? There are different methods of adding. It could be directly added like so, or it could be added using a delivery guy called as vector, about which you are going to learn in detail again. All right. So now I have a few hosts, same organism, many copies of the same organism, into which I have tried my level best to insert my gene of interest. But how do I know whether it has got inserted or not? I can't tell. Looking at it, I can't tell. So we need to have a checking mechanism, quality control. Okay. So that quality control checking mechanism is called as selectable marker, which is going to be a little tough. You don't understand it, but you will understand. I'll try to make it simple. All right. So once I check for the recombinants or transformants, okay, transformants. What does that mean? The host organism which has taken up my gene of interest. The ones who have not taken up, we are going to call as non-transformants, and the ones that we have, the ones that has taken up, we are going to call as transformants. Not transformers, transformants. All right. What do I do with this transformants? I need to make it work. I need this organism to make lot of copies of the protein that I need from the gene of interest. So this gene, every gene, transcribes and translates finally into giving you some protein, isn't it? So for this purpose, I'm going to use some methods. Either I am going to do a small scale production of producing this protein in the lab, or I'm going to do large scale production using bioreactors. So this entire chapter is all these. However, in our NCRT book, they have divided all these into different groups, that is tools and processes. But it is better we learn in this sequence. So let me try to make you understand each of these steps in detail in the upcoming videos. Thank you.